In the last video, I introduced the concept of evolution, and we talked about traits and the requirement of traits to be heritable, meaning they have a genetic basis to them. And we talked about how traits become adaptations, how they meet the challenges that all animals have to face from an ecological perspective. The traits that become better adapted in those situations, become adaptations to face those challenges, are molded by a process of natural selection. And remember the three requirements of natural selection, the first of which is there has to be variation among individuals in the population with regard to the trait that you're talking about. The second requirement is that that trait is heritable. And then the third requirement is that some traits have better fitness than others. So we introduced the concept of fitness. But remember that first requirement is that there's variation. And that variation we talked about comes from mutations. And we talked about the operation of mutation at the genetic level. So that's how organisms evolve in a microevolutionary sense. And this lecture is going to focus on macroevolution and how we build phylogenies to both inform our classifications, but then also how we use phylogenies to track the evolution of traits of interest, to see how traits have evolved as different lineages face different ecological challenges. What are the major traits that they have in a macroevolutionary sense? So since the 1700s, we've used a system developed by Carolus Linnaeus to classify all living organisms. And it's a hierarchical classification system, meaning that there are like boxes within boxes. There are groups that the membership criteria are relatively broad. It doesn't take much, for example, to be an animal. But then you get into more specific animal groups. So the kingdom Animalia is the biggest group. But then you have different phyla. The plural of phylum is phyla. So you could have the chordates, the chordata, and that would include us. It would include leopard frogs. But something like a katydid, that's in a different phylum. That's in the Arthropoda. And as you go down this list, we go from inclusive groups to more and more exclusive groups. So the subphylum has fewer representatives and the criteria for membership are more strict compared to a phylum. And then a class is more restrictive still, down to order, family, until we get down to species. And the criteria for membership to be in a species is the most specific. So here's kind of a graphical way of looking at that. So again, in this biggest box, we have a kingdom. And in this kingdom, there may be, say, 3 phyla. And within each phylum, we could have a couple of classes, or in some case, one class. So that's one important thing here. These don't have to be balanced. They're based upon the degrees of similarities and the criteria of the traits that are shared among members of each of these groups. So in some cases, there may be multiple families within an order, and in some cases, there may only be one family within an order. And we've used this Linnaean classification system for a very long time. Well, I mentioned that the species was the most basic unit to describe a basic kind of organism. It's the most specific of the taxonomic units in the Linnaean hierarchy. So what do we mean by a species, or what do we mean by a separate kind of organism? Well, in general, I think you know what this means. So if you go outside and you see a bird, you see a northern mockingbird, that's the state bird of Texas, you say, well, that's a different kind of bird than a northern cardinal. Those are different kinds of birds. Those are different species. The characteristics of species are they have to be genetically or morphologically distinct groups of individuals. So, I mean, if you look at a cardinal, again, you can see that 3 different cardinals are going to look more similar to each other than either of them is going to look to another species of bird. This is because they share a common descent from an ancestral population. They look the same because they have received the same genes from their ancestors. They belong to a lineage that shares the same gene pool. They keep their gene pool together and separate from other groups of organisms because they can breed with each other, but they can't breed outside of their species typically. And that's what gives us this kind of natural distinction for what a species is. Well, one of the things I want to make sure that you know is how to write a species name. A species name is represented by a Latin binomial, a two-word name. So here are a couple of examples. This bird on the right, the common name for this bird in most English books is the blue-tailed bee-eater. But that's not a good species name to use in science, because if you travel to different places and different languages, they could use completely different names. So, for example, this bird in Mandarin Chinese is Li Ho Feng Fu, and in Thai, it's Nok Leong. So we need a common language that we can communicate, and so we've decided that using a scientific Latin binomial is the way to go. So in this case, this bird is Merops philippinus. You are a Homo sapien. That's what humans are as a species. Well, let's break down this name and understand what the different components of it are. This is a bird that I caught in Taiwan. The common name is the hoopoe, and the scientific name is Upupa epops, one of my favorite scientific names. So notice first the italics in the pattern of capitalization. The species name is italicized to set it off from the rest of the text that you're looking at. So it basically says, here you're looking at a species name. You're looking at something that's written in Latin. All right, notice also that the first part of the name, Upupa in this case, is capitalized, but the second word is not capitalized. This first word represents what we call the genus. The second word represents what we call the specific epithet. And you would never use the specific epithet alone. So epops by itself doesn't mean anything. There are a lot of specific epithets like Jamaicaensis. Okay, Buteo jamaicaensis refers to a red-tailed hawk. But if you just say Jamaicaensis, there are probably hundreds of species names that end with that specific epithet, but they don't mean anything until you link them to the genus you're talking about. So a specific species name is really only properly indicated when you're using both parts of the name together. So Upupa epops is the hoopoe. Now, if you're writing a species name and you can't, if you're writing in cursive or you're printing and you can't, to make it stand out in a written statement, what you would do is you would underline the species name. And you would actually underline the genus and then leave a space and then underline the specific epithet. So I mentioned that we've used the Linnaean classification system for quite a while. But now we're moving to using phylogenetic relationships and using phylogenetic trees to develop modern classifications and kind of leave off the terms like kingdom and phylum and class and order and just talk about clades. A clade is a branching group that starts from a common ancestor and includes all of its descendants. So one of the clades shown here in red is the arthropods. If you look from this branch, this orange branch that includes this orange branch and the red ones, that is the Panarthropoda clade. And then outside of that, if you look at this kind of mustard colored one here, we have an even more inclusive clade, the Ecdysozoa. So it's using the same basic principles the Linnaean classification did. It's boxes within boxes. It's groups within groups. But now we're looking at it in a structured phylogenetic manner. And this is the phylogeny we're going to be covering and using throughout the semester to track animal diversity. How we build these phylogenies and the characters that we trace or we use to look at their history of development or evolution across this phylogeny, we use character homology. Okay, so a character that's homologous is a character that's shared among organisms due to it being inherited from a common ancestor. So what we're saying is that there are characteristics that all arthropods have, homologous traits, because they inherited it from their common ancestor at this common branch. There are characteristics that all chordates have, that they've inherited from their common ancestor. Same thing for the ambulacra. Same thing for all deuterostomes. So we use character homology to build this group within group branching pattern in a phylogeny. So let's give you some examples of homologies. One of the classic examples is the pentadactylous limbs of tetrapods, so 5-finger and 4-limbed organisms. If you look at those, they actually serve a huge diversity of functions. So we've got legs in frogs, very different from the legs in horses, which are very different from the flippers in a porpoise, and very different again from the wing of a bat and the grasping arm of a human. But if you look at these traits, despite the fact that they have very different functions, they are clearly homologous. They came from a common ancestor because they have the same basic underlying parts. So they all have a humerus. They all have a radius and an ulna. They have carpals, metacarpals, and phalanges as colored here. That only makes sense if they've inherited the basic genetic framework to produce these basic structures from their common ancestor. Now, why do they look different? Remember, we talked about pleiotropic developmental genes and how they can change the relative shapes and sizes of structures. They all have the genes that say make a humerus, but different developmental genes have said make a humerus in a different way and have it articulate in a different way with the radius and the ulna. If you look at within a group like birds, you see that there are certain things that they all share together. They all have this high degree of fusion in the bones of the hand, or what we refer to as the manus. They got that because they inherited it from, again, a common ancestor of all birds. This indicates this shared ancestry of all birds. So this specific type of homology we call a shared derived homology. So all of the organisms share this derived condition, this changed shape. Birds have this great degree of fusion because a common ancestor of all birds first derived that change. Now there's another term for a shared derived homology. It's also called a synapomorphy. And if you break that word down, you see that it really does mean the same thing. So homology, remember, refers to like a structure. That's referring to morphes here. Apo means change. Okay, that's the derived part. Syn means together. So it's a shared, changed morphology, shared derived homology. So again, if we look in this phylogeny, if we didn't know how these branches really fit together, but we had a character that said E, F, G, and H share this very same trait. Well, the most logical inference is the reason they share that same trait is they got it from their common ancestor. It was a shared derived homology, or a synapomorphy. So this group we would then call a monophyletic group, or a clade. So shared derived homologies or synapomorphies are lines of evidence that a group is unique somehow, that it forms a monophyletic group. Okay, let's break down that word, monophyletic. Phyletic refers to like a branching pattern. Mono means that branching pattern comes from a single origin. Okay, so that's what we're looking for. When we're making our phylogenies, we're trying to discover shared derived homologies as evidence of what groups we can put together as monophyletic groups. So again, here would be a shared derived homology that would support that as a monophyletic group. That's our goal in building phylogenies, and that's the logic of phylogenetic biology. Now there's another way of drawing phylogenetic trees. Instead of drawing them kind of like in these bracket forms, sometimes they're drawn in triangles that fit within each other. I kind of like this form better, but the one on the left is typically more commonly seen in papers and books. So again, let's go through some examples of synapomorphies or shared derived homologies and the clades or monophyletic groups that they support. Again, if you just had these organisms up here, an Amphioxus, a bass, a lizard, a horse, and a monkey, you'd say, okay, well, what are some of the characters that I can use to decide which of these organisms are more closely related to each other? Well, if you said, what about hair? Well, horses have hair, monkeys have hair, these others don't have hair. They also have mammary glands. Okay, those are shared traits that were derived in the common ancestor of all mammals. So that is a synapomorphy for this box right here. Horses and monkeys make a monophyletic group that we call the mammals. All right, well, what about the other organisms? Are there traits that we can use to try to figure out how they're more closely related or distantly related to horses and monkeys? Well, a lizard has 4 legs and uses in its reproduction amniotic eggs, which is a big deal, as we'll see later in the semester. This is going to eventually tell you the answer to the old question, what came first, the chicken or the egg? And I'm telling you right now, it's the egg. Phylogenetic biology tells us that. Anyway, okay, to continue. So, for the presence of 4 legs in the descendants of this common ancestor, and the descendants also come from an ancestor that had amniotic eggs, again, these are shared derived homologies or synapomorphies supporting the lizard, the horse, and the monkey, representing a clade that we call the tetrapods. And then the next, out of these 2, the next group, the bass, the remaining animal here that clearly is a vertebrate. It has vertebra and jaws just like these others do, but it lacks these traits. So that would be a synapomorphy for the vertebrates. So that's really our goal. We're trying to come up with classifications that accurately reflect past evolutionary patterns based upon monophyletic groups being supported by shared derived homologies or synapomorphies. Those are the types of characters that we want to use. But sometimes we get characters that, because of the rate at which they evolve or because of the circumstances in which they evolve, they can kind of mislead us. And we want to try to avoid those. So again, we're trying to look for synapomorphies to support something like this monophyletic group. We don't want a trait that seems to indicate that C through H belong to each other more closely, and A and B don't. Because C, really what you're doing, C and D, if this is the true branching structure, C and D don't really belong with these guys over here, they really belong with A and B. So if you ended up forming a group like that, that's a paraphyletic group. We don't want to do that. Over here is even a worse example, a worse mistake that you could make. If we put the clade E, F, and G, which is a good group, but say they actually belong really close related to C and D, see, they're not even connected by a blue branch. That is what we call a polyphyletic group. We don't want to do that either. So let's look at the types of characters and why they might mislead us in those circumstances. So again, we want to do monophyletic groups, clades based on shared derived homologies or synapomorphies. This paraphyletic group could be formed if you paid attention to traits that are called shared ancestral homologies. This is when you almost form a correct group, but you leave out some of the descendants. So a paraphyletic group is the ancestor and some of the descendants, but not all of the descendants. You've left these guys out for some reason. In this case, if you paid attention to this trait right here, it's called a shared ancestral homology or a simplesiomorphy. If you looked at a character, for example, that said C through H belong together to the exclusion of A and B, you'd be making a paraphyletic group. But really what's happened is whatever this character is, it's the ancestral form that has simply evolved past that in this group that has a faster rate of evolution. So, for example, the traditional group we call the reptiles excluded birds. Well, birds are reptiles. They just don't look very reptilian in many of their characteristics. They've evolved wings. They've kind of evolved past that ancestral state. So if you use those ancestral traits, you'd be using shared ancestral homologies to say lizards and dinosaurs, non-avian dinosaurs, and snakes and turtles. Those all belong to the group, the reptiles, to the exclusion of birds. And that would be wrong, because that as a group reptiles defined that way is not a monophyletic group, it's a paraphyletic group because it leaves out, in this case, A and B, which in this example I'm using is birds. Now, if you found characteristics that said, okay, evolved here and was found in all of them, sure, that's a monophyletic group, right? Because it includes everything. But if you just use a shared ancestral homology that has some of these but not others, then you'd be making a mistake. Another term for a shared ancestral homology is a sympplesiomorphy. Again, morphy refers to some trait. Plesio means old or ancestral, and sim is kind of like syn and synapomorphy. It means together. So it's you're mistakenly grouping a group together based upon some ancestral trait. A shared ancestral homology shared by some, but not all of the descendants. So we don't want to do that, and we also don't want to make polyphyletic groups. Polyphyletic groups are put together when you are comparing apples and oranges when you think you really just have two sets of apples. The characters in this case are what we call homoplasies. These are characters that are not due to homology. They're not due to common ancestry. They just kind of look the same because they've evolved through a process of something like convergent evolution. So we may have a trait here and here that are seeing E, F, and G, and also C and D, and you think, oh, well, you know, I see. So here's would be an example of this. Bats have wings. Birds have wings. But you wouldn't say that those wings are really the same thing. If you say that birds and bats belong together, you'd be making a polyphyletic group because the bats here would go with the other mammals, right? And the birds here would go with the other reptiles. So that would be making a big mistake. You would be forming a polyphyletic group, and we don't want to do that. So here's some classic examples of traits that might at first mislead you. If you look at flippers and fins in aquatic organisms, is this really an indication of their close relatives? In other words, would you say that these are synapomorphies supporting them belonging in a clade? Well, hopefully you're saying no, right? Because this is a bird. A penguin is a bird. That is really just a highly derived wing. That is an ancestral fin that you see in fishes. In this case, it's a cartilaginous fish. And then the flipper of a dolphin or porpoise, again, that's a completely different structure that's derived from an ancestor that used terrestrial tetrapod legs. So on the outside, these things look similar, but if you really understand how these were produced, you know that you wouldn't use that character. And you may be thinking, well, how am I going to know that? How do you know which traits you can trust and which you don't? Well, think about phylogenetics. The way it works is if you're comparing a whole bunch of different characters, most of them are going to agree on a certain pattern, and then something else is going to stick out like a sore thumb. So, in this case, there are lots of characteristics of a penguin, like the presence of feathers and their internal anatomy, and that differ from that of a shark. And then there are characteristics, a lot of characteristics that indicate that this is a mammal. So it's just this one character, the fin here, that you might superficially think puts them together, but there are a lot of other signals that say, no, that's a bird, that's a fish, and that's a mammal. I said that the reason this similarity occurs is through a process called convergent evolution. So they have converged on the same solution for the same environmental challenge. If you're going to swim, a flipper-like structure works well. And so that's evolved independently in each of these groups. That's what it means to have convergent evolution. They've converged on the same basic trait as a solution to a similar ecological problem. So again, this semester we're going to track the evolution of characters based on this phylogeny. And this phylogeny is based primarily now on genetic similarity, genetic changes that we can use to understand which of these groups are clades based upon changes in the DNA. So there are lots and lots. If you look at similar DNA strands, you can see shared derived homologies that indicate where these branches should go. And some of the characteristics we're going to track early on are going to be some of the most basic fundamental aspects of an animal, how complex it is, whether it has no germ tissues, just a series of tissues glued together, of cells glued together, as in a sponge, or if they're diploblastic with endoderm and ectoderm, or if they have endoderm, ectoderm, and sandwiched in between that, mesoderm. And then we'll also talk about, how that mesoderm produces a interior cavity, and how that differs between different lineages of organisms. So that previous figure, that phylogeny, is going to be a more fine-tuned way of looking at this basic pattern shown in this summary pattern of animal evolution. So in review, we talked about the traditional hierarchical classifications developed by Linnaeus, and we talked about the specific, one of the specific groups within that category, the species, and how species really are kind of naturally self-defining because of their interbreeding and sharing of a common gene pool and making it distinct from other species' gene pools. We talked about how you write a species name. It's a Latin binomial. Make sure that you know the different parts and how to write those parts correctly, the genus and the specific epithet. And then we talked about how we're really shifting away from the Linnaean system and moving to a more phylogeny-based classification system derived from the study of homologies, specifically shared derived homologies or synapomorphies that help us understand monophyletic groups, but how we also can use patterns of other characters' evolution on properly formatted phylogenies based on shared derived homologies to understand things like shared ancestral homologies. Why are birds different in so many of their characters? Because of the simplesiomorphies that are found in non-avian reptiles. And why that sometimes leads us to, in the past, make paraphyletic groups. And also in the past, we have sometimes formed using mistaken homoplasies for homologies, and this has led to the formation of polyphyletic groups. But this is due to patterns of convergent evolution, and actually understanding when convergent evolution happens is actually interesting in itself because it tells us a lot about adaptive evolution and what traits serve what function in common environmental circumstances. Okay, in the next series of videos, we're going to start our exploration of the evolution of the basic anatomy and physiology of animals. And we're going to start by just talking about some of the basic body plans and levels of organization and how that varies among animals.